So let's start discussing the formed elements of blood. As I said, red blood cells, white blood cells, and platelets. Red blood cells are also called erythrocytes. White blood cells are also called leukocytes, and platelets are platelets. Okay, so let's talk about erythrocytes first off in this video. Erythrocytes are also called red blood cells. Just so you know, the lifespan of a red blood cell is about 120 days, about 4 months. They are anucleate, which means there is no nucleus. And additionally, there are very few organelles present. The main function of an erythrocyte is to transport hemoglobin. This makes up about 97% of its molecular content. I would like you to look up the numbers for a typical red blood cell count. There are varying numbers depending on where you look, but we'll talk about some specific numbers in class. Okay, so if our the main function of red blood cells is to transport hemoglobin, we should discuss that next. So here is hemoglobin. Let us discuss the structure of hemoglobin. It is a protein called globin and a pigment called heme. And more specifically, there are 4 globins and 4 hemes. You should be able to readily recognize this structure whenever you see it. It's extremely important. When we look at a typical protein, it oftentimes kind of looks like just a bunch of stuff twisted together because the shape of the function of proteins is that they are twisty and take on a particular shape. So in this case, though, I want you to recognize that there are 4 subunits here. There's this kind of weird thing here, back here, here, and here. These are what we call globin chains. There are alpha chains and beta chains. Not super important to recognize one from the other. Okay, so these are called polypeptide chains, which peptide is a peptide bond is what holds the amino acids together. And so 4 polypeptide chain basically means that each of these are subunits of a much larger structure called a protein. Okay, inside of each of the globins then, there's 1, 2, 3, 4 globins, and what you'll see is that there is that red heme group inside of there. Let's take a closer look at that. This is what a heme actually looks like. It does tend to take on a red color, especially when it is bound to a molecule of oxygen. So a heme pigment or a heme ring bears 1 iron atom, Fe, right in the center here, and that's very important. Each iron can bind to 1 molecule of oxygen, which always exists as an O2. And just so you know, 1 red blood cell can carry 250 million hemoglobins, and we're going to play around with those numbers a little bit. But I want you to think about how many total molecules of oxygen a single red blood cell can carry. See if you can figure that out and calculate it out and come to class with that number. Okay. Let's take a look at the process called erythropoiesis. There's the spelling right up at the top there. Erythropoiesis is the formation of red blood cells. As I said, they last about 4 months. They last about 120 days, but they must be regenerated all the time because by the time you have a mature erythrocyte, it will never manufacture another one. It does not undergo mitosis, and in fact, it is not actually a living cell. The erythrocyte, as you can see here, does not have a nucleus. So we're going to start off this entire process here. It's not important to know all of the little individual names of all of the intermediaries here, but we do have to know the name of this structure here. It's called a hemocytoblast. It's what we call the hematopoietic stem cell that eventually differentiates into a red blood cell. Okay, so erythropoiesis is an extraordinary feat that the body conducts. We create about 2 million or more new red blood cells per second, which indicates that they are very important to us. The process occurs in the red bone marrow. It takes 3 to 5 days to become this second to last stage here, which is a reticulocyte. And it takes about 2 days for the reticulocyte to mature into an erythrocyte. Now, a reticulocyte actually takes on some of the roles of a mature erythrocyte. In other words, it does begin to transport oxygen. Reticulocytes account for about 1 to 2% of all the erythrocytes that you'll find in the bloodstream. All right, the reason why a hemocytoblast would begin to undergo the process of erythropoiesis and eventually become an erythrocyte is because of the presence of a hormone called EPO, or erythropoietin. If you look at the last 3 letters here, SIS indicates that it is a process. The hormone that causes the process to go forward is called erythropoietin, so you just have to change these last 3 letters from cis to tin. Okay, erythropoietin is a hormone that is produced within the kidneys, and there's a number of reasons why erythropoietin might be released by the kidneys, and all of them have to do with the idea that you need more red blood cells. Since the primary role of an erythrocyte is to carry oxygen, I want you to think about different reasons why it is that EPO, erythropoietin, might be released from the kidneys. So under certain circumstances, we release more erythropoietin, and why would that ever be useful? And then the second part of this is I want you to consider why it is that erythropoietin, EPO, might be abused by athletes.